It is day one of the JP Morgan Healthcare Conference. I'm here in the Flagship studio, joined by Ted Myles, who joined Cellarity as CEO about 6 months ago, and also Flagship as a CEO partner. Ted, Cellarity has become a clinical stage company in the last year, and with one program, and you have a second one in the hopper. I want to talk about both. Yeah. But let's start. You presented today on the big show, the main stage. What was your main message for investors in that presentation?
Couple main messages. Message one, we are past the question, can AI be used to assist, enable, fuel R&D? Celarity's been leading this charge for a long time. The company was founded in 2019 with this mission, and we are now in the clinic, and we're about a year away from demonstrating proof of concept with a molecule that was not only created based on this AI platform, but the target was actually discovered. It was new biology that was discovered because of this AI platform. So that was one big message. AI is here for real. Celarity is a leader in it.
Great.
And CLY-124, our sickle program, is in clinic and about a year from proving our thesis that we, hopefully, knock wood.
That's terrific. There's been a lot of talk in the industry over the last several years about can AI really deliver. You guys are one of the first companies to be doing that and in the clinic. So tell me a little bit more about CLY-124. It's in sickle. What is different about this molecule? Because there are many players in sickle cell disease. What should we be watching for this year, and why is this molecule different than others?
Yeah, so sickle cell's been an area of intense focus for the biotech industry. I'd say we as an industry have not quite delivered for patients the way we had hoped. There's been a lot of promise. There's really two big buckets. There's those that induce fetal hemoglobin and all the other approaches. Within the fetal hemoglobin inducers, you've really got gene therapy and the oral inducers. The primary example being standard of care hydroxyurea. The gene therapy has produced some miraculous results for a few patients. The challenge is accessibility from a price standpoint. Administration's very hard. You have to go through a toxic regimen to go through it. It's incredibly expensive and incredibly time consuming. So it's really limited the uptake. And so scientifically, it was a success. Commercially and real impact for patients, it's been very limited. On the oral fetal inducer side, we've really seen there's hydroxyurea, which is standard of care. It increases fetal hemoglobin to about 11%, which is symptom reducing, but not curative. The reason they can't dose up further is because it runs in a cytotoxic pathway. It actually forces adult hemoglobin to become fetal hemoglobin, stresses the bone marrow. Novel approach, again, we could not have discovered this biology with any other traditional method, so it was really the Cellarity platform that allowed us to uncover this biology. We talk about discover, create together at Cellarity, discover new biology that hasn't been seen previously, create novel chemical entities to interrogate that biology, and we do it by bringing together this broad swath of R&D capabilities from biology, computational biology, chemistry, and of course AI, ML. So
you talked about 11% in terms of where current oral therapies can get in terms of inducing fetal hemoglobin. What preclinical data do you have that gave you confidence to move this program into the clinic, and where does that benchmark against that 11%?
Great question. So there's two elements to the preclinical data to give us confidence. I'll touch on cytopenia first. So we tested our tool compound equivalent of CLY-124 at target dose and 30 times intended human dose, and really tried to force cell death and couldn't do it. And we compared that to all the other oral fetal inducers, and it's 70 to 80% cell death. It's really, really significant. So that was safety, and that was that confirmation of a novel pathway. Because we induce fetal hemoglobin before the cell matures, so there's no bone marrow stress like there is with the other approaches. Then there's, okay, it's safe, great, does it work? Right? And so we did a number of experiments to show that we use a humanized mouse model. There's a gold standard mouse model. We use CLY-124. And we started to see ranges of 20 to 40% fetal hemoglobin. Very widely accepted bar out there that above 20% is a functional cure.
So more than double current standard of care, and maybe
significantly more than double current standard of care without, hopefully, the dose-limiting toxicities. Right. So safely, high tolerance, no need for constant monitoring. that's the dream. If we can deliver what we sometimes refer to as gene therapy-like efficacy in a once-a-day pill.
In a pill.
Yeah.
So good preclinical data. What should we be looking for in 2026 with this program? You mentioned proof of concept data.
Yeah, so even before we get there, you mentioned we entered the clinic. So
Yep.
we started dosing subjects, healthy volunteers, in June. Happy to say we've now dosed over 70 healthy volunteers safely at dose escalating from a tiny
Wow.
15 mgs up to 1,200 mgs. We've learned that it's well tolerated. There have been no SAEs, there have been no SUSARs, and really importantly, the target engagement of this new biology, this new pathway that's never been explored, which is called DCN1, the target engagement exceeded our expectations.
Fantastic.
Which means that we're starting to hone in on our sickle cell dose, which we're starting imminently at 200 milligrams.
Okay.
And so we feel very good about that we're getting nice engagement, and we should see a signal
So
in
70 healthy
2026.
volunteers about to start in sickle patients.
Correct.
And then you'll see that first signal towards the end of this year.
Possibly. So one of the really big, really great breakthrough through the development and regulatory organization, and then we had a great board meeting about this and aligned on what I think is a pretty powerful path where we're running our multiple ascending dose cohorts and we're running parallel cohorts. So we're going
Okay.
to test this molecule as a monotherapy. That's the intent. We believe CLY-124 should be a monotherapy curative for all patients. But it's important to test patients who are on hydroxyurea. So we have approval to do that in Kenya. There's a lot of sickle cell disease in Kenya. We've got our eyes set on a couple of sites that have a lot of patients on hydroxyurea. So testing in mono on one cohort, one set of cohorts, and in combo on another set of
Okay.
cohorts is going to A, help enrollment, B, we think we've got a synergistic effect with hydroxyurea, which again is more evidence that it's a completely different mechanism. We're not stacking impact on top of itself. We could see a signal much earlier.
Okay.
We'll see. It depends what dose we're on, depends where the patients are. But importantly, we can get to the other end of this experiment and see that it works with hydroxyurea, great patient benefit there, and it works as a monotherapy. So when we think about commercially, again, curative for all patients. And so physician adoption, they won't have to take a patient off of standard of care
Yep.
to try a new therapy. Right? We're thinking about this from a very long-term perspective.
Myelofibrosis is up next. Tell me about that program within the context of cell state correcting.
It's been really fascinating. The myelofibrosis program, from the time I joined, we would hear a bit about it. It felt like it was a little stuck in the mud. We finally leaned in, gave it a little bit of budget, and all of a sudden here we are. We've got some really interesting in vitro data, and it gave us conviction to run a really exciting in vivo experiment where we're going to go head-to-head against the standard of care. And what we're going to try to demonstrate is that we not only can improve symptoms, but we can do it in a very safe way. So the standard of care right now has been a remarkably successful therapy, symptom reducing, but it's got a lot of side effects.
Okay.
This is a disease that's really in need of a lot of innovation. It's a multi-billion-dollar product, even with some spleen reduction, some other symptomatic reduction, not curative, and this very tough side effect profile. What it's challenged in doing is separating or differentiating between healthy cells and sick cells in the bone marrow. It just sort of wipes out everything. Novel mechanism of action. We utilized our platform. We discovered new biology. We have this new target that we're interrogating. We created now a series of chemical entities. We're kind of figuring out which is our lead, and we're about to start these in vivo studies to show, hopefully, that we can have same or better impact on the symptoms, but also separate. And the in vitro work we did showed that we can really suppress the aberrant cells and spare the healthy cells.
Okay.
So that the patients can get better, right, get rid of the toxic mutant cells, keep the healthy cells in the bone marrow so they can continue to make red blood cells, white blood cells, platelets.
So only
that's
targeting the
critical.
bad, keeping the good.
Targeting the bad, keep
Yep.
it good.
Great. So both of those programs are wholly owned?
Wholly owned,
Okay.
created within the platform.
And tell us about your BD strategy, because there's other things in the pipeline or things that you could do with the platform. How are you thinking about that?
Yeah, this is one of those. It's a blessing and a curse, right? We have a platform. I don't think we've even begun to explore the width or depth of this platform. So there's no scenario where we can do everything on our own, nor should we. So BD is a really important part of our strategy. About 3 years ago, the company signed a deal with Novo Nordisk, working in MASH. That was at completely ground zero. They just had an idea, and we've been working with them all along.
And that's a liver disease, right?
That's a liver disease, and
so that would take you
in the
into a
metabolic
different
area.
Yeah.
So we're supporting their initiative. This is one example of a BD transaction where we would work with the partner, explore what's interesting to them, and utilize our platform and our expertise to help solve their issue. Another version of a good BD strategy that we're employing, where we've already done some work. We've created some cell atlases and some cell maps, and we're looking at an early hypothesis. And we just signed a deal in Q4 with our affiliates at Pioneering Medicines. And we're looking at how our T-cell strategy can work in inflammatory bowel disease.
Oh, interesting.
So
a couple different categories and different ways to point the platform.
What I really like is we can do a lot of these other programs and start to define ourselves. We can be, our core can be in hematology, and with sickle and MF and a few other things in the hopper that we're evaluating. And then immune, there's so much white space there that we can work with partners in different forms of partnership. And then, of course, in metabolic, we could just be scratching the surface with Novo.
Neat.
Yeah.
So let me close with a little bit of a personal question, if you'll indulge me. You have been at this conference many, many times, wearing many different hats.
Yeah.
An operator, a chief financial officer, private company, public company.
Yeah.
What feels special or different about this moment, both for you and maybe, I don't know, for the industry or for Cellarity, but you're coming at it for the first time as CEO?
Yeah.
What are your thoughts?
Well, so whenever you ask anyone about the JP Morgan conference, the first reaction is the weather. The weather's good, so it's going to be a great year in biotech. I think that's scientifically proven. I was just explaining to a colleague that every JP Morgan seems to have its own flavor. And if you're with a team that you really respect and enjoy working with, and you've got a company you're really proud of, whatever the backdrop is, you can usually make a pretty good conference out of it.
Yeah.
I say that we have that in spades. We're in a really good position right now. I haven't been here as in a private company setting in a very long time, but it's all the same physics. It's all about, proving, laying out a plan, figuring out, yes, we can improve patient care, laying out a set of experiments that will help de-risk that program in the eyes of investors and partners, and telling the world what we're going to do.
Great.
And then doing it so that we can come back next year and,
say you did it.
Further along. Yeah.
Well, we will absolutely be watching. Ted, thank you for joining me here in the studio.
Thank you.